Cell culture is one of the major techniques in the life sciences. The most important and crucial step in cell culture is choosing an appropriate growth medium. A typical culture medium consists of a complement of amino acids, vitamins, inorganic salts, glucose, and serum as a source of growth factors, hormones, and attachment factors. In addition to nutrients, the medium also helps maintain pH and osmolality. Animal cells can be cultured either using a completely natural medium or an artificial/synthetic medium along with some natural products. Natural media consists solely of naturally occurring biological fluids. Natural media are very useful and convenient for a wide range of animal cell culture. The major disadvantage of natural media is its poor reproducibility due to lack of knowledge of the exact composition of these natural media. Artificial or synthetic media are prepared by adding nutrients, vitamins, salts, O2 and CO2 gas phases, serum proteins, carbohydrates, cofactors. Different artificial media have been devised to serve one or more of the following purposes: 1. Immediate survival, 2. Prolonged survival, 3. Indefinite growth, 4. Specialized functions. Artificial media are grouped into 4 categories: serum-containing media, serum-free media, chemically defined media, and protein-free media. Serum-containing media. Fetal bovine serum is the most common supplement in animal cell culture media. It is used as a low-cost supplement to provide an optimal culture medium. Serum provides carriers or chelators for labile or water-insoluble nutrients, hormones and growth factors, protease inhibitors, and binds and neutralizes toxic moieties. Serum-free media. The presence of serum in the media has many drawbacks and can lead to serious misinterpretations in immunological studies. A number of serum-free media have been developed. These media are generally specifically formulated to support the culture of a single cell type, and incorporate defined quantities of purified growth factors, lipoproteins, and other proteins, which are otherwise usually provided by the serum. Chemically defined media. These media contain contamination-free ultra-pure inorganic and organic ingredients, and may also contain pure protein additives, like growth factors. Their constituents are produced in bacteria or yeast by genetic engineering with the addition of vitamins, cholesterol, specific amino acids, and fatty acids. Protein-free media. Protein-free media do not contain any protein and only contain non-protein constituents. Compared to serum-supplemented media, use of protein-free media promotes superior cell growth and protein expression and facilitates downstream purification of any expressed product. Basic components of culture media. Culture media contain a mixture of amino acids, glucose, salts, vitamins, and other nutrients, and available either as a powder or as a liquid form from commercial suppliers. The requirements for these components vary among cell lines, and these differences are partly responsible for the extensive number of medium formulations. Each component performs a specific function, as described below. Buffering systems. Regulating pH is critical for optimum culture conditions and is generally achieved by one of the 2 buffering systems. Natural buffering system. A natural buffering system is low cost and non-toxic. HEPIs. HEPIs is relatively expensive and toxic at a higher concentration for some cell types. Phenol red. Most of the commercially available culture media include phenol red as a pH indicator, which allows constant monitoring of pH. Inorganic salt. Inorganic salt in the media helps to retain the osmotic balance and help in regulating membrane potential by providing sodium, potassium, and calcium ions. Amino acids are the building blocks of proteins, and thus are obligatory ingredients of all known cell culture media. Essential amino acids must be included in the culture media as cells cannot synthesize these by themselves. They are required for the proliferation of cells and their concentration determines the maximum achievable cell density. L-glutamine, an essential amino acid, is particularly important. Non-essential amino acids may also be added to the medium to replace those that have been depleted during growth. Supplementation of media with non-essential amino acids stimulates growth and prolongs the viability of the cells. Carbohydrates. Carbohydrates in the form of sugars are the major source of energy. Most of the media contain glucose and galactose, however, some contain maltose and fructose. Proteins and peptides. The most commonly used proteins and peptides are albumin, transferrin, and fibronectin. They are particularly important in serum-free media. Fatty acids and lipids. They are particularly important in serum-free media as they are generally present in serum. Vitamins. Vitamins cannot be synthesized in sufficient quantities by cells and are therefore important supplements required in tissue culture. Trace elements. Trace elements are often supplemented to serum-free media to replace those normally found in serum. These micronutrients are essential for many biological processes, for example, the maintenance of the functionality of enzymes. Media supplements. The complete growth media recommended for certain cell lines requires additional components which are not present in the basal media and serum. These components, supplements, help sustain proliferation and maintain normal cell metabolism. The shelf life of the growth media changes after the addition of supplements. Complete media containing protein supplement tend to degrade faster than basal media alone. Antibiotics. Although not required for cell growth, antibiotics are often used to control the growth of bacterial and fungal contaminants. Routine use of antibiotics for cell culture is not recommended since antibiotics can mask contamination by mycoplasma and resistant bacteria. Moreover, antibiotics can also interfere with the metabolism of sensitive cells. Serum is a complex mix of albumins, growth factors and growth inhibitors. Serum is one of the most important components of cell culture media and serves as a source for amino acids, proteins, vitamins, particularly fat-soluble vitamins such as A, D, E, and K, carbohydrates, lipids, hormones, growth factors, minerals, and trace elements. Serum from fetal and calf bovine sources are commonly used to support the growth of cells in culture. Due to the presence of both growth factors and inhibitors, the role of serum in cell culture is very complex. Table 1 shows the advantages and disadvantages of using serum in the media. Criteria for selecting media. The choice of cell culture media is extremely important, and significantly affects the success of cell culture experiments. The selection of the media depends on the type of cells to be cultured and also the purpose of the culture and resources available in the laboratory. Different cell types have highly specific growth requirements, therefore, the most suitable media for each cell type must be determined experimentally. In general, it's always good to start with MEM for adherent cells and RPMI 1640 for suspension cells. Table 2 describes commonly studied cell lines and recommended growth media. Primary cell culture provides unique and valuable research data, but most of the time cell number is the limitation. For such critical samples, especially from diseased human biopsies, a quality medium is required. Creative Bioarray provides complete and ready-to-use, fully supplemented conditioned medium. This reduces the risk of contamination as well as save time, labor and money by eliminating the preparation steps and supplementation required. Moreover, all of these media are subjected to comprehensive quality control tests and each lot is routinely tested for growth promotion, the absence of cytotoxicity, and physical parameters such as osmolality and pH level. Thanks for listening. If you have any questions about cell culture, please feel free to contact us in the following ways.